All right, so we want to thank you all, both online and here in person, for coming to our first joint biology chemistry assembly for this year. Last year we had one, which was actually our very first one. And part of the reason for doing this is to focus on the fact that many of our problems that we have today could only be solved by disciplines coming together, interdisciplinary, multidisciplinary approaches. And that's what you will find with our speaker today. Let me I know all of you should have received the email and you already have the bio of the speaker, so I'll be very brief and give her most of the time. So our speaker, Professor Claudia Fischbach, and the last name, how do you pronounce that correctly?
Teschl. Teschl.
Teschl, okay.
It's an L at the end.
Okay, Teschl. She's the Professor of Biomedical Engineering at Cornell University. She received her Ph.D. in Pharmaceutical Technology from the University of Regensburg in Germany and conducted postdoctoral work at Harvard University in the Division of Engineering and Applied Sciences. So today she will focus on tumor engineering and cancer. And oftentimes we do not put the word cancer together with engineering. And so you will see a really interesting approach to solving the problems of cancer. Thank you again, Professor, for being with us and accepting our invitation.
Well, thank you so much for inviting me. It's my pleasure, and I'm completely with you. I think interdisciplinary work is very important, and I usually don't start my talk this way, but just to give you a little bit of background, I'm actually a pharmacist by training, so I'm not an engineer, and I'm not, biologist by training either. But what fascinated me back in the days about pharmacy, there was a lot of chemistry. And there was a lot of engineering principles, and I was interested in applying them to studying biological questions. That was basically the combination. And while at the time, I oftentimes felt like I'm neither here nor there, so I know a little bit of everything, but nothing as much in detail as, for example, a chemist would or a biologist would. I think what it has allowed me over the years is actually be able to communicate to both audiences in a way that it has helped bring our research together. And biomedical engineering intrinsically is an interdisciplinary field where different disciplines work together. So I am located on the Ithaca campus of Cornell. When I give seminars to more biologically inspired people, they often think of Weill Cornell Medicine in New York City. And yes, we're one university, but we're apart, 4 to 5 hours, depending on which time of the day you're going. And yet we're actually closely collaborating regardless. So what I'm going to do today is tell you about our work that we've done in tumor engineering. And presumably not all of you are thinking, hopefully not thinking about cancer on a day-to-day basis because it's affecting you and your families, and probably not from a research perspective either. So I would like to start with an analogy that I like using to explain how we're approaching this idea of cancer and why it's developing. And all of, as you know, all of us have cells in our body that function abnormally. And there's various control mechanisms in place to keep these cells under control. And the way I like describing them is, yes, there's intrinsic mechanisms that you probably have heard about, but cancer cells are like a seed that need the right soil to grow. Only if the surrounding of the tumor cells, not only the tumor cells themselves, but if their surrounding is permissive, then that cell can take root and can grow. And what's happening during the progression of tumors, once they've developed, then they actually further modulate their environment in a way that it becomes even more permissive, and you're basically, you know, activating this positive feedback loop of tumor development and progression. And while we know this, and while the field knows this, there's still most of the current cancer research and therapies focus on the tumor cells themselves and largely disregard what that soil surrounding the tumor cells is. And that's not completely correct anymore, that statement, because if you think of immunotherapy, that is the first approach that really targets the microenvironment or the soil rather than the tumor cells themselves. Anyway, what I also like using is this example of Angelina Jolie, just to emphasize the importance of, you know, thinking beyond genetics, because most of you are probably familiar with her case. She's a carrier for BRCA1 mutation, which significantly increases her risk to develop either breast or ovarian cancer, and therefore she had both of these tissues removed. And yet, even though that mutation is one of the few that's known to increase dramatically the risk for cancer development, many of BRCA1 mutation carriers never develop cancer. And vice versa, 93% of all breast cancers actually can't be traced back to initial gene mutations. Over time, every cancer becomes a genetic disease, but whether or not it's the initial driver for tumor development is actually not as well established. So how do we think about that soil or the microenvironment? It's basically an aspect that we should be considering across all stages of the disease. So initially, the tumor develops in primarily epithelial tissues. These epithelial cells lose their polarity, start growing. They eventually start invading into the surrounding normal tissue. They can enter blood vessels in this process called intravasation. They travel through our system, and then at distant sites, they're extravasating and then forming metastasis. And in all epithelial-derived cancers, the metastases are typically the case where patient prognosis decreases dramatically. So because we know that the microenvironment is important, there's lots of interest in studying those connections. Other cells, growth factors, cytokines, vesicles, etc. They are all obviously part of how that microenvironment or that soil is composed. But what people oftentimes disregard or forget is that there is also that extracellular matrix that is basically the glue in between the cells, and that also kind of functions almost as like a storage material for growth factors and cytokines. And yet we know very little about the extracell matrix. The extracell matrix at a molecular level is obviously composed by proteins. There is sugar-based components, glycans, proteoglycans, but also, as I just said, there's soluble factors that are integrated into that extracell matrix. But at a more, mundane level, matrix is basically a material. And that material is not only determined, or the let's say biophysical properties of that material are not only determined by its biochemical composition, but also by its structure, and as a consequence, then also by its mechanical properties. So it's really a material, and you can look at it from a material science perspective and characterize it that way. And even though we know that, matrix, again, is not a typical focus of cancer research. If you just do a search for publications, entering keywords for tumor genetics or tumor extracell matrix, you find a huge discrepancy between these two areas. So we know very, very little about what matrix does, and that is in part because traditionally tumor cells have been studied in petri dishes or cell culture dishes on plastic, or using mouse models primarily. And yet, if you obviously you realize that plastic is not the best representative of our matrix in the body, and vice versa. In animal models, this is a collagen-stained histological cross-section of human breast tissue. The red is collagen, and this is what the same tissue looks like in a mouse. So again, mammary gland in a mouse versus human, same magnification, same stain, taken by the same person at the same time. And you can appreciate that these two look very different. So really trying to think about how can we better mimic some of those scenarios where tumor cells interact with matrix, and then control and understand those scenarios. And that's basically where the engineering comes in, and what my lab is doing. We're using synthetic and natural biomaterials as artificial extracellular matrices, and we fabricate them in different sizes, shapes, consistencies, with and without microfluidic conduits or patterns, to basically design what we call biomimetic tumor microenvironments that then allow us to address or answer specific questions that we're interested in. And there's several areas in our lab, but one of the, or actually I would like to introduce two areas from our lab, and one focuses on adipose tissue. You might be wondering why adipose tissue, and the interest simply comes from the fact that many, or perhaps even most, of the epithelial-derived cancers, they're residing in locations that are rich in adipose tissue. So breast tissue is a prime example. A large component, or the primary stromal component of breast tissue, is fat, or is adipose tissue. But the same is also true for colorectal cancer, ovarian cancer, prostate cancer. All of these cancers are basically embedded within fat tissue or adipose tissue. And tumor cells, actually, as they're invading right in between those cells that are characteristic of adipose tissue. Those are the fat cells, the large black holes here. So when you look at adipose tissue in a histological cross-section, this is an H&E stained cross-section, you appreciate these cells are very, very big. Those are adipocytes, and they're simply big because they store so much lipid. In between are what we call stromal cells, and in particular, in between the adipocytes, there are so-called adipose stromal cells that in part have stem cell properties, and it's those stem cell properties that allow these adipose stromal cells to self-renew and to differentiate into more adipocytes, typically as there's, for example, an excess in energy supply. But what you can also see is this is what adipose tissue looks like in the presence of a tumor. So all of this is tumor. And again, this is adipose tissue, but you see that the tumor cells are invading that adipose tissue, and now it looks quite different. And so when I first started my lab, we were simply interested in the question, okay, how do these adipose stromal cells change in the presence of a tumor? And what my very first PhD student, Emily Brooks, did, she collected conditioned media from a number of different cell lines and primary tumor cells, and then applied it to adipose progenitor cells, or adipose stromal cells, and basically looked at whether they still have the ability to develop into fat cells. And interestingly enough, when these adipose progenitor cells were fed with tumor conditioned media, we didn't see any incorporation of lipid droplets anymore, which in the control conditions are the red stained nodules here. We didn't see activation of this transcription factor PPAR gamma anymore that you would expect in the nucleus. But instead, what these cells did is they didn't develop into adipocytes anymore, as I just told you, but they upregulated markers that are consistent with myofibroblasts. And that marker is oftentimes, or alpha smooth muscle actin is oftentimes used as a marker of these myofibroblastic cells. And these myofibroblastic cells are cells that in your daily life, for example, as you're cooking and you're burning yourself, or perhaps you're ironing a shirt and you're burning yourself, or you're cutting yourself, there's a wound healing process that is activated. And it's those myofibroblastic cells that during normal wound healing processes start depositing matrix. They deposit fibronectin, collagen, so much more mesenchymal types of matrix components relative to what epithelial cells will do. And they start aligning that matrix and making the matrix stiffer. And that is what you see later on in scar tissue formation. And that those matrix changes during normal wound healing are necessary because they basically provide epithelial cells that contribute to wound healing with a guidance scaffold that allows these cells to grow in and then basically heal your wound. In tumors, those myofibroblasts do something very similar. However, they don't stop producing that abundant amount of matrix and remodeling that matrix during tumor progression, but they do more and more and more. And this is eventually what can be palpated. This is why people oftentimes detect tumors themselves, or a physician can do it. Or also those changes is actually, or those changes are what typical clinical imaging methods are picking up, like mammography, for example, or also MRI. They're looking for tissue changes in tissue density that are primarily mediated by those exocell matrix changes. So anyway, what Emily did is, now that she knew these adipose progenitor cells don't develop into adipocytes anymore, she tested whether they actually deposit matrix that is similar to a tumor-associated matrix. And yes, these cells did make more collagen and fibronectin. They were contractile. So this is a collagen contraction assay where the adipose progenitor cells fed with tumor-conditioned media collected from MDA and B231 cells. Those collagen discs contracted. You see that the fibers that are deposited are much more aligned and longer. And at a molecular level, we could show together with Delphine Godin that these fibers partially unfolded, and that all of these things together actually contributed to increased stiffness of matrix deposited by adipose progenitor cells in the presence of tumor-conditioned media relative to control cells. So was this relevant? Yes, it was, because what Emily then went on to show was that if she's implanting tumor cells alone, they have tumor-associated matrix associated with them. But if she implanted tumor cells along with adipose progenitor cells, now that matrix deposition was much more abundant, the fibers were more aligned, and the tumors were stiffer. All of these things contributed to making the tumor grow faster, to be more vascularized, and to be more aggressive. So that was interesting. And basically, I tell you about this data, even though they were collected quite a number of years ago, because they motivated a lot of research that is still ongoing in our lab, and that is collected in the context of obesity, where we know that obesity is obviously on the rise, not only in the US, but worldwide. I think America is a leader. But the problem with obesity is obviously huge problems in cardiovascular and metabolic diseases. But also we know that it significantly increases the risk for cancer development and worsens the prognosis of patients that already have cancers or tumors, because obesity increases the aggressiveness of these tumors. So we were wondering at the time, so, well, if adipose tissue is important in tumor progression, as I just showed you, what if obesity is an intrinsic modulator of adipose tissue, and could that in part explain these numbers of worse prognosis and higher risk? And so Bori was a student in my lab who followed up in this and basically worked with colleagues at the medical school and at Sloan Kettering Cancer Center, using a combination of diet-induced mouse models of obesity or genetically induced mouse models of obesity, as well as patient samples. And what she found, that all of these changes that I just described to you in the context of tumors are already initiated by obesity alone. So in the absence of a tumor, obese adipose tissue, where you not only see now these adipocytes become significantly larger, right? You see that relative to adipose tissue from mice, but the matrix in between those adipocytes is increased for collagen, as stained here with picroceres as red. Second harmonic generation imaging shows you that these collagen fibers are not, curly and wavy anymore, but now they're actually aligned. And that alignment, again, is characteristic of increased tissue stiffness or matrix stiffness. And yes, of course, if you now measure stiffness in those matrix areas, we found that obesity increases this interstitial matrix stiffness. And again, that is important because we know that stiffness in and of itself can actually be a driver of increased tumor cell proliferation per se. So what Boris was interested in is like figuring out, okay, is it indeed these cells, these adipose progenitor cells or stromal cells that I told you about that are responsible for those changes because they might be changing with obesity? And yes, indeed, that was the case. So what she did was she isolated these adipose stromal cells either from lean mice or from obese mice, again, diet-induced or genetically induced obesity. And what she found was that with obesity, alpha smooth muscle actin, this myofibroblastic marker, was upregulated. These cells deposited more collagen, they deposited more fibronectin, they structured the matrix differentially. Again, they aligned fibers, they unfolded fibers. And then again, with Delphine, when we measured the mechanical stiffness of that matrix that was deposited by obese stromal cells relative to lean stromal cells, that was significantly stiffer. So now we know that, okay, these adipose progenitor cells are key regulators of tumorigenesis in normal adipose tissue, but they change with obesity. But how can we now look at whether or not these changes are actually altering cell behavior as a consequence? And the way we're doing this in the lab is, again, using a combination of synthetic materials, but also cell-derived matrices that we basically collect by letting adipose stromal cells deposit matrix in regular tissue culture. Then we get rid of the adipose stromal cells, so we decellularize those matrices that the cells have deposited, and then we replate whichever cell type we're interested in, for example, the tumor cells, onto the matrix, and then can study their behavior. So those cell-derived matrices, these ones here, we've characterized them via proteomics, and you see that there's key regulators of fibrotic matrix remodeling that are upregulated, present in those matrices. But over the years, what we have now done, use those substrates, again, a combination of these decellularized matrices and more synthetically defined matrices, to basically show that the matrix, the compositional and structurally and mechanically induced changes of matrix by obesity not only activate tumor cells to grow more and invade more, but they basically change the cellular composition and the cellular phenotypes in the whole microenvironment. For example, we have shown that collagen structural changes, I'm sorry, this is induced by obesity, they themselves, independent of any biochemical factors, promote myofibroblast differentiation. And that, of course, if you have even more of the scarring response, and we know that it is driving tumorigenesis, then that obviously is a worse predictor for tumor development and progression. But what we also found was that macrophages, non-polarized macrophages, respond to these changes in matrix biophysical properties and basically assume phenotypes that are consistent with tumor-associated macrophages. So basically suggesting that the matrix is a material that, independent of soluble factor signaling, can activate a whole bunch of changes in the tumor microenvironment that we know is contributing to tumor progression. And what I'll do is I will actually skip these next slides because I want to make sure we have enough time, and also then enough time to answer questions. But basically, what the conclusions of that first part of my talk is that adipose tissue is important, matrix is important in adipose tissue, and that obesity can alter the mechanical properties of mammary adipose tissue by basically promoting profibrotic matrix remodeling. But also what we do know is that some of these adipose stromal cell contractility changes directly promote invasion as well. So invasion obviously is the initial driver of tumor cells dislodging from their primary tumor and undergoing key steps necessary for metastasis to occur. And this is also invasion is the driver of when a tumor transitions from being benign to becoming malignant. And this is what a pathologist would look at. If somebody has to have a mass removed, they are basically looking at the boundary of that mass and look at, okay, is it still very locally confined, or has it started to progress and invade into the surrounding tissue? And with engineering approaches and matrix models, including advanced imaging methods and computational simulations. And even though I didn't talk about it, but those things are really key to figuring out, okay, what are the mechanisms that tumor cells use to interact with that matrix and invade eventually. But as I just told you, in most of the cases, in almost all of the cancers, other than, of course, brain cancer and lung cancer, patients don't die from primary breast cancer. They can have, their breasts removed. There's therapies that actually function quite effectively. However, once the tumor has metastasized, then that is where, clinical prognosis decreases dramatically. And in the case of metastatic breast cancer patients, the primary organ or tissue that is being targeted by metastatic breast cancer cells is the skeleton. And in the skeleton, the tumors basically activate processes that are very similar to osteoporosis. They degrade the bone. There's a pathological fracture. These patients have a huge amount of pain. There's hypercalcemia, and eventually the prognosis of these individuals is very poor. And because of those obviously dramatic consequences, there's a lot of interest in figuring out, and we know that tumor cells secrete factors that activate the bone degrading cells, the osteoclasts, and inhibit the bone forming cells, which are the osteoblasts. And so you see all of these relatively complex interactions between receptors and signaling factors. But, just remember what our skeleton does in addition to providing, calcium homeostasis. It's basically a biomechanical entity that allows us to walk and to jump and do all of the things that we need to do in our daily lives. And yet, even though we understand bone matrix is a driver of all of this, we know very little about how bone matrix is regulating metastasis formation. And while obviously it is really important to think about, okay, how can we treat and improve prognosis of a patient who already has metastasis? I think what is perhaps the even more promising approach is to intervene at stages where we know the tumor cells are already in the skeleton but haven't formed a metastasis yet. Because what is happening is that dissemination, which is the spreading to distant organs, actually happens very, very early during disease progression, sometimes even before a primary tumor can be detected. What is interesting about this dissemination is that not every tumor cell that goes somewhere ultimately develops metastasis. And we know this because people in autopsy patients that haven't died from cancer, but where these patients may have had cancer at some point in their life, these patients have disseminated tumor cells in their skeleton, for example. And so the big question of the field is, why can it be that tumor cells go to certain locations and can hang out there for years or decades, and sometimes forever, and in other cases, they become activated, and what drives their activation? Because if we understood those processes, we could intervene and can basically prevent metastasis formation rather than try to cure something that is almost impossible to cure. So what is interesting about these very early stages of bone metastasis is that tumor cells target regions in which new bone is formed rather than degrade it. So as I told you before, once metastasis is clinically detectable, the bone degrades as during osteoporosis. However, in these very, very early stages, the tumor cells actually go to what's called osteogenic niches, where, again, new bone is formed. And new bone formation is happening in our skeleton all the time. For example, as you're exercising or as you're growing, your bones need to deposit new bone constantly to become strong. And we know that during this initial stage of tumor dissemination, tumor cells interact with the bone-forming cells, these osteoblasts, and people have described cell-cell interactions be involved in this process. But one of the key functions of these osteoblasts, hallmark function, is that they make new matrix, new bone matrix. And the way they make matrix by first depositing collagen fibrils. And these collagen fibrils then over time become mineralized. And that is what's the defining, obviously, biomechanical entity of our skeleton, those collagen, mineral incorporating collagen fibrils. And yet we don't know, do tumor cells, once they disseminate to the skeleton or to these osteogenic regions, do they actually interact with mineralizing bone matrix? And the reason we don't know this until recently, or until we've started to look into this, is because it's really difficult to look at bone mineralization using conventional techniques, because you're interested in these early stages in very, very, very small numbers of tumor cells within the bone. So you need to be able to go in at high resolution. And if you're interested in their interactions with mineralization, you can't use demineralized bones because obviously you would get rid of all of the mineral. However, in order to do histology, you can't just cut a bone because of the mineral, but you first need to demineralize it, and then you can send it off to histology so that they can section the tissue and you can look at its cellular composition. So basically, you can pick either or. And however, there is good techniques where you can label mineralizing bone matrix. But the problem with those is that typically these strategies are applied to PMMA embedded bones, and this PMMA embedding is a very harsh process. And so if you're using cells that are labeled for example, with a fluorescent protein, all of that fluorescence would be gone. So you can basically either image the bone matrix, or you can image the fluorescence of the cells. And so we tackled this challenge, or more directly, Aaron Cho, who was a PhD student in my lab, and Josh Hinckley, a PhD student in Uli Wiesner's lab, with whom we collaborated. And we wanted to label the cells with a fluorophore that is very stable under harsh processing conditions that allows us to maintain the mineral. And so the approach we chose was to use silicon nanoparticles that Uli's lab is developing. These silicon nanoparticles contain synthetic dyes that are super bright. They are very, very stable, and they're biocompatible. Uli's lab actually uses these particles for clinical imaging. So they're applied in patients in clinical trials right now. And what Aaron did was he basically just let the cells take up those particles and showed, yes, the particles are inside the cells. And then he used those labeled cells and injected them into mice in which he previously injected calcine, which is this molecule that basically incorporates with a newly mineralizing bone matrix. And then after one day, because this is the day where following injection, all of the non-adhered cells have cleared out of the bone, and only the ones that would seed there would be left. So he injected the tumor cells. After they took out the bone, optically cleared the bone, and subjected these bones to light sheet imaging, which basically allows us to look throughout the whole bone without sectioning the bone. And this is what this looks like. So I'll show you a video in a second, but basically the green is where new mineralized matrix has been deposited following calcine injection. So this is a 1-day time frame that you're looking at here, and it's pretty remarkable how much of our bone is constantly remodeled. Well, this is a mouse, by the way, so I shouldn't say our bone. And then the pink stain is labeling the tumor cells. You see some autofluorescence here, but those are not individual cells. But we can also detect individual cells as they're being localized within the marrow. So we're now going into the bone, and you can see where these individual tumor cells are located. And then you can basically generate optical sections and look in more detail, okay, where is newly mineralizing bone in green, and where do our tumor cells locate? And so, of course, there's some in the marrow, as you would expect, right? But there's also some that directly interact with the green regions, which is the mineralizing region. So that basically provided some justification for us to look at this in more detail. And again, I have to come back to this idea of, okay, why should we worry about bone mineral matrix? It is this hydroxyapatite or mineral reinforced collagen composite material that at its smallest scale is basically determining what our bone tissue is in terms of its biomechanical properties. What is interesting is that we know that bone mineral density or bone matrix or formation of bone mineral matrix decreases with age, but also decreases, for example, in response to chemotherapy. And epidemiologically, people have found that patients that have reduced bone mineral density have a higher risk of developing bone metastasis. So we were then questioning this idea if perhaps bone matrix, so physiological bone matrix mineralization or maintenance of physiological bone matrix mineralization increases latency of tumor cells, so keeps the tumor cells basically in this non-growing state, while reduced bone matrix mineralization activates the tumor cells to grow. And so the way we tried to address this question is taking advantage of Xiang's skills. He's a material scientist, postdoc in my lab, and he developed a collagen system, a collagen scaffold system, in which he deposited collagen fibers and mineralized these fibers, either conventional methods that people use to just mineralize surfaces such orthotopic or prosthetic implants, where you're depositing mineral on top of surfaces, or using a physiological process where you're actually mineralizing collagen intrafibrillarly. And the way this works in our bones is that collagen has this ultrastructure with gap regions where the fibrils are assembling. And mineralization in our bone matrix happens in these gap regions of the collagen. Eventually, all fibers get wrapped by mineral, and that is determining, you know, the compressive strength, but also the tensile strength of our bone, which are very unique, and again allow us to do all of the cool things that we can typically do. Xiang synthesized these materials. He characterized them using FTIR to ensure, yes, we have collagen there, and yes, we have a hydroxyapatite there. And then he's also ensured that this mineralization increases the stiffness of these materials, as you would expect also to happen in our bones. And what he then did is he cultured tumor cells on those different substrates and did RNA sequencing. And principal component analysis is typically done just to see, okay, do cells on the two different substrate systems actually cluster differently in their gene expression profile? And yes, they do. Mineralized collagen cells are very different in their gene expression from cells cultured on collagen. And when you look at what is differentially regulated on these two substrates, what was interesting using gene set enrichment analysis, that a lot of the biological processes that showed up were somehow related to DNA replication, proliferation, mitosis, etc., when cells were cultured on collagen. In contrast, cells cultured on mineralized collagen had changes in their metabolic activities, matrix disassembly. But because we were interested in this question of dormancy or latency, we followed up and said, well, if there was more proliferation in collagen, let's look at actual proliferation markers. And sure enough, when we looked at, for example, BRDU incorporation, which is a proliferation marker, cells proliferated significantly less on mineralized collagen relative to collagen. And that was not only true for one cell line, but we tested this for multiple cell lines, including this bone metastatic subclone of MDA and B231 breast cancer cells. So this is the parental cells. This is the bone metastatic subclone. And this is another breast cancer cell that represents estrogen receptor positive breast cancer, which is most frequent, the most frequent subtype of breast cancer in postmenopausal women. So this was interesting because as I told you before, but not in as much detail, we know that increased matrix stiffness is known to be promoting malignant phenotype. So there was a landmark paper published by Valerie Weaver in 2005 that was just right around when, you know, I was in my postdoc and like thinking about faculty positions. And what this paper showed was that an increase in matrix stiffness can promote tumor cell proliferation independent of any other things. And this increase in stiffness, again, is primarily mediated by increased matrix density and collagen density. And the way cells activate cell proliferation in response to stiffness is they do this because they can spread more on stiffer materials. The spreading leads to a change in how cells assemble their adhesions. And then this change in adhesion assembly formation activates downstream signaling, such as phosphorylation of focal adhesion kinase on the Y397 residue. And that is then activating myosin contractility and activation of a bunch of transcription factors involved in proliferation. This increase in cell spreading and in integrin adhesion formation also allows the cells to actually adhere more strongly. You can measure this with traction force microscopy. And so basically with increased matrix rigidity, not only do the cells spread more, but they exert stronger forces at those adhesions. And that is something that people have now taken as, you know, is a relatively established concept in the meantime, that we know, okay, increased stiffness promotes tumor cell proliferation and risk of developing cancer. This is why, for example, in New York State, women who do go and get a mammogram and have dense breast tissue and therefore stiffer breast tissue, that they are being sent home with a note to say, you have dense breast tissue, you had increased risk to develop breast cancer. So now this was interesting because we actually saw on the mineralized collagen the opposite behavior. So the mineralization, as I told you before, increases matrix stiffness, and yet you clearly see that the cells on that stiffer matrix spread less relative to the softer collagen matrix. So opposite of what I just told you about stiffness. Also, we saw that mineralized collagen resulted in a decrease at focal adhesion kinase phosphorylation at the Y397 residue. Correspondingly, phosphomyosin light chain phosphorylation, which is an indicator of actomyosin contractility, decreased, and traction forces of the cells cultured on the mineralized collagen decreased as well relative to cells cultured on collagen. And so that was interesting because, again, it is opposite of what we think to know about stiffness. And the way it can be explained is that a lot of the stiffness experiments are done with materials that are linearly elastic, which means it deforms at a linear rate as cells exert forces. However, and that basically that deformation is then enforcing the adhesions. However, collagen is not a purely elastic material, and this is where the engineering perhaps comes in, but it's a viscoelastic material. So in which the stress-strain response is not happening in a linear manner. And that is because collagen can undergo what's called local strain stiffening. So as cells adhere and interact with collagen fibers, what they do is they actually, and this is confocal reflectance imaging from a paper from Peter Friedel's lab that I like showing, but I think this schematic here is even nicer. The integrins engage, they interact with individual collagen fibers, but there's this positive feedback loop that the force they're exerting onto those fibers actually leads to the local alignment of fibers in those regions close to the adhesions. And you see that here by this increase in the red color. And that densification and alignment of fibers actually leads to local strain stiffening, which you can measure with AFM mapping. So this is an AFM map. This is where the adhesions are, and this is basically where the local strain stiffening of the collagen is occurring. And so what we think is happening with collagen mineralization is that the mineralization, even though it increases compressive stiffness of the material, it actually prevents the cells to align the collagen fibers, but they're now basically stuck in place because of the mineralization. And that doesn't allow these cells to form stronger adhesions, which reinforce the collagen fiber alignment. And this is the pseudo-coloring of the surrounding collagen. You see the collagen density around those cells adhering on collagen locally increases, whereas this is very little the case on the mineralized collagen. And sure enough, consistently, if we're interfering with cell contractility, which should be increased with local collagen alignment and densification, has an effect on the growth of cells on collagen, but not on mineralized collagen. We basically, added a number of either function inhibiting antibodies or pharmacological inhibitors of downstream signaling of downstream mechanosignaling. And again, this suggested that mechanosignaling is increased on the softer collagen relative to mineralized collagen. It has something to do with the inability of the tumor cells to remodel that mineralized collagen. So that is interesting, right? Because again, that would support this idea that collagen mineralization is actually really important, and therefore bone matrix health is really important for perhaps maintaining quiescence or latency of tumor cells in our skeleton following their dissemination. But then, of course, you can argue, well, this collagen system is so artificial. What, the matrix in our bones is much more complex, and there's other proteins, even though to a lower extent. There's other structures, perhaps. And so to validate the importance of mineral, Matt Whitman, PhD student in my lab, he basically used bone plugs that he harvested from bovine femurs. He removed all of the marrow. He removed all of the cells. And then he used those residual scaffolds that he had to either in their native state, so with the mineral, but no cells, no marrow, or he now further demineralized and got rid of the mineral. And that only left the organic matrix. And what you see is that in SEM, but we've also looked at it at the collagen fibrillar levels, all of the ultrastructure was maintained. The only thing that differed was the amount of mineral present in these two scaffold systems. And sure enough, also in those models, the presence of mineral decreased proliferation of tumor cells relative to scaffolds in which the mineral was lacking. The nice thing about these scaffolds is also that you can now use them for in vivo studies where you can implant the scaffolds following seeding with breast cancer cells, and then you can follow, okay, how do tumor cells grow, either in the presence of this mineralized bone matrix, which is red here, or in the presence of demineralized bone matrix. And clearly what you see is that tumor cells grew significantly more when the mineral was absent. So in these demineralized or scaffolds without mineral. So the other nice thing that we can do with this data, or actually let me step back. So the combination of these in vitro data that I just showed you and the in vivo data that are depicted here basically suggest that the tumor cells assume phenotypes in the presence of bone mineralization that keeps them in a less active, less proliferative state, and that might be contributing to latency.
So, Professor, could you give us maybe a couple of minutes conclusion and stuff, because we're going to lose our audience here pretty quickly.
Yeah, I have 2 more slides.
Okay.
So, but what is nice about the gene expression data is that you can now overlay them over patient data and basically make predictions about whether or not the presence of mineral activates genes that are associated with worse or better prognosis. And sure enough, that tumor cells in patients now that express high levels of a gene expression score induced by mineral fared overall clinically much better than patients that expressed low levels of these mineral inducible genes. And this correlation was more important in patients with luminal breast cancer, which metastasized to bone much more frequently to basal cases. In those basal cases, the tumors basically metastasize everywhere. So that also supports this connection as being relevant. All right, so basically what we think is happening here is that following dissemination to bone, tumor cells interact with newly mineralizing bone matrix, and that mineralization is critically important for them to maintain their less proliferative state, more latent state, and that we need advanced imaging modalities and model systems to basically figure out the underlying signaling and use that in the future in the context of patient outcome. So with that, I, of course, need to appreciate all of my students and collaborators and funding sources that were involved with this work. I'm fortunate to work with a really tremendous group of people. And I'm happy to answer any questions if people are still around.
Okay.
Thank you.
Some are around still, yes. Okay, thank you very much. Let's give a round of applause. So you guys have two options. Those of you who are on Zoom, you can put your questions in the chat. Are there any questions in the room? Okay. A question that I have is your transition from pharmaceutical technology, without too much biology, you said as well, into this work that you're currently doing. What was that like? What got you interested in this type of research?
Well, pharmaceutical technology is a broad field, right? What I did for my PhD was basically tissue engineering work. And that's where pharmaceutical technology, drug delivered biomaterials, is really a field that involves an understanding of material properties, exploring material properties for delivering molecules to a patient. And that has expanded into delivering cells and cellular systems into patients, and then further expanded into this idea of, okay, developing model systems to better understand biology and potentially also use for drug testing applications. So there's a logical connection between a lot of these things. And if you look at pharmaceutical, industry more broadly, I think, or in whichever students take their undergraduate classes and get bachelor's degrees, but then evolve into very different areas as they're entering the workforce. And so I think that also is something that happened to me in this process.
All right, sounds good. Yeah, we are kind of losing our audience in here now, but we want to go ahead and thank you. Unless there is somebody else that has a question online. We want to go ahead and thank you very much for your presentation and you continued success in your work.
Thank you very much. I appreciate the invitation, and good luck to you as well.
Okay, thanks. Take care. All right, bye-bye. Bye-bye.